So welcome everybody to the first webinar of the VALITEST webinar series on the organization of test performance studies. So VALITEST is an EU-funded project built to improve the reliability of diagnostic tests performed in plant health laboratories across the European and Mediterranean region. The project is undertaken by a consortium of 16 partners composed of research institutions, private companies, NPPOs, and intergovernmental organizations. And it has three main objectives: to provide a more complete and precise description of the performance of diagnostic tests, to stimulate and optimize and strengthen the interaction between stakeholders in plant health for better diagnostics, and to lay the foundation for structuring the quality and the commercial offers for plant health diagnostic tools. So we already organized a first webinar series on the concept of test validation in plant health, and this is our second webinar series, which is on the organization of test performance studies. Test performance studies are interlaboratory comparison that aims at evaluating the performance of diagnostic tests. And during this series, VALITEST partners will present the different steps of the TPS workflow through different videos, webinars, and training activities, practical training activities. So a first video was released yesterday on what is a TPS, and another video will be released today on the selection of PEST and TPS organizers for VALITEST TPS. But during today's webinar, we will present the different elements that should be considered when planning a TPS. So just to let you know that this presentation will be recorded, and a link to the recording and the PDF of the presentation will be provided on the VALITEST website. So our speakers today, Françoise, if you can switch to the next slide. So our first speaker is Françoise Peter. So Françoise is the Deputy Head of the European and Mediterranean Plant Protection Organization. And for more than 15 years, she has been in charge of the EPO program on diagnostics and is the chairman of the EPO panels on diagnostic. I will also present part of this webinar. So I am a scientific officer at EPO since August 2019, and I am working on the VALITEST project and on other EPO diagnostic activities. And at the end of the webinar, three TPS organizers will join us for the Q&A session. So Natasha Mel, who works at the National Institute of Biology in Slovenia, and who organized the TPS on tomato spotted wilt virus. Then Monica Mezzalama, who works at the University of Torino in Italy and organized the TPS on Cryphonectria parasitica. And finally, Katrina Harrison, who works at FERA in the UK and organized the TPS on Fusarium circinatum. So with no further ado, I will give the floor to Françoise.
Good morning, everybody. So we would also like with Charlotte to acknowledge the contribution of Géraldine Antoine, who is unfortunately not able to be the presenter for this webinar, but we had preliminary discussion with her, and she had a great contribution to this presentation as well. So what will we be speaking about? So we will introduce TPS versus proficiency test, and then we will speak a little bit more about the TPS plan together with Charlotte. Charlotte has organized interviews during the project with TPS organizers, so you will have a movie and with the interviews, and at the end we will have a question and answer session, and I'm really grateful to the TPS organizers to be with us today to be able to answer the practical question that you I'm sure will have on test performance studies. So the inter-laboratory comparison, as Charlotte explained, are of two types. And they are test performance studies that are also called ring test or collaborative trials. You find that quite often in publications, and proficiency tests. And these inter-laboratory comparison have really been very important in plant health. They have become an important part of the process, in addition to the validation process that may be done on intra-laboratory level. And I would also like to insist on the small video that tells you all about what are interlaboratory comparisons. So if you have young colleagues coming in your lab, please show them the video, because in a nutshell, they will understand the difference and the objectives. So because these were becoming more and more important, we were asked by experts in the region, laboratory diagnosticians, if we could not try to help with harmonization of the organization of this interlaboratory comparison. So harmonization, and at the same time provide guidance to those who want to start with an interlaboratory comparison. And this is the reason why we started in the year 2012, if I remember correctly, to develop the guidelines on the organization of the interlaboratory comparison. And our standard was published in 2014, and it has been used as a basis for VALITEST. And one of our big hope is, and we know it's going to happen, is that based on the experience of this, many different test performance studies organized in the framework of that project, we will be able to revise our standard to make even better to serve the needs of the countries. So just shortly, test performance study is the evaluation of the performance of the test. Proficiency test is the evaluation of the performance of a laboratory. So they are different objectives for PTs and TPS. They are explained in the standard PM7 122. And here in this slide, we are only presenting the main objective. You can have other, some PTs may also help to give information on tests. Some TPS may also give information on proficiency. But here, what is presented are the main objectives. And you can see that there is a difference. And we will be focusing on test performance studies. And here what you're looking at is comparability of test, effectiveness of a test, establishment of uncertainty levels, and evaluation of performance characteristic of a test, which is really important for having an idea about how a test behaves. So we will start with our first poll, because we'd like to know more about you who are listening to this webinar. So who are you? Do you organize inter-laboratory comparison? If no, just answer, yes, I do test performance study. Yes, I do proficiency test. Yes, I do both. So please answer the poll.
So about 90% of the people have answered. I will wait a few more seconds. Yeah, it's not changing. So I will close the poll and share the results. So we have about 37% of the people that are not organizing interlaboratory comparison. 19% only TPS, 19% only proficiency test, and 26% both.
Okay, so I have a plea to those who are organizing test performance studies. If you have anything to contribute at the end of the presentation, please feel free to do it, because it's really great to have contribution. You will have the panelists, you will hear from the interviews, but if you could really contribute, and because that's also the objective, that we share experience, because the more we share experience, the better we can prepare documents to help others. So don't hesitate to contribute. So as explained, our focus will be on test performance studies today. So if you look at a TPS plan, in fact, it has different elements. How you plan your TPS. So you're interested in the capacity, in your own capacity to organize a TPS. So are you able to organize a TPS? What is your scope, including which test you want to include in your TPS? So the participants who will take part of your TPS, the material you will need, the sample preparation and dispatch, the analysis of the result, and then how do I disseminate and communicate about my results? So that's quite that will be the different elements that we will browse today with you. And what you have to know is that for each of these steps, when you prepare your plan, what is important is that you keep in mind that you should identify the major sources of error at each step. Because when you identify the sources, you can also mitigate for these problems. So the first thing is the capacity of the organizer. So as organizer of a TPS, you need to have the equipment, the facilities, the experience, and if you don't have it, the question that comes is, maybe I could subcontract. So is it possible to subcontract for a TPS? In fact, not for the technical competence and the tests. So you need to be proficient on the tests that you will do your TPS on and on the tests. And we had the this experience in one of the TPS where a lung test could have been introduced in the TPS, but the organizer said, I don't have the competence in that test. So this was a test that was not selected. The only subcontracting activity that is possible is a statistical analysis. There's one point I forgot to mention in this graphical presentation that we have here. Maybe it's a little bit unfortunate that we use a kind of fishbone representation, but what we want to show here are the different elements to consider. So don't think there is any chronology or whatever there. It's just looking at all the elements that you need to take into account. So you have the equipment and facilities, you have the experience. You also need to look at, do I have my procedure and do I have sufficient forms to run the process? Do I have the resources? So do I have enough people to perform this activity? And in particular, if I also want to be part of the TPS, how should I organize it? So you should not have a single person. You should really think about how you can organize this. And of course, what is really important is that you need a clear designation of your responsible staff. So regarding basically as an organizer, basically you really need to have a quality assurance system in place in your laboratory, and these are described in two other EPO standards. We're not going to go into detail on that, but you need to have appropriate equipment and facilities to prepare the samples and handle them and dispatch them. And this could also be the case for the reagent. You have to ensure that you don't have any cross-contamination between the test samples, because that could really seriously jeopardize the outcome. So in your lab, you need to monitor the environmental condition. You need clear separation of areas in your laboratory. The equipment required, we saw it in the first slide, and the necessary action to handle quarantine pests, because in our work in EPO, we are mainly considering quarantine pass. So if you do it on a quarantine pass, you need to have the appropriate facility to handle quarantine pests or take the measures like inactivation, etc. So the first thing is the scope of your TPS. And clearly, when you start a TPS, for sure, you already know when you decide to do a TPS, you know why and what for you're going to do it. So what for? Basically, why performing a TPS? So they are tests that are used by the NPPO in their surveillance, and you see that there is really a need for more validation data. So your objective would be to gather more validation data on tests that are currently used for surveillance. Also, you have done a validation intra-lab, and you want to have more information on your test and how it behaves by having it evaluated in multiple laboratories. So that's important, and that will show you elements regarding more information about reproducibility of your test. Yeah, and sometimes you really want to know how your test will behave. The pest could be, do I want to do it at a higher level than the species? So this will have an influence on what you do in your TPS. Do you want to do it at subspecies level? So you really need to define very clearly what you want to do and which pest is there. What are the hosts of the pest? Which metrics do you want to evaluate? What will be the intended use of your test? So do you want to use a test that do you want to evaluate a test that can be used at field level, so the on-site test? How many control will I have to include in my TPS? The regulation or standards, it might be the case that there is a regulation that imposes a number of tests. For example, in the European Union, there have been for a long time control directive where tests are specified. And so organizing a test performance study, in this case, you have to use the tests that are included in the regulation. So you don't have that much choice. Regarding the organization itself, you can look at, are there any other standards available to help me with that? Or is there an EPO standard on diagnostic from which I could see which tests could be useful to evaluate? Do I intend to do it at national level or at regional level, or do I see big? So I want really to have it at international level. And so what could be the consequences? The first one, and when we were discussing this presentation, one of the points Charlotte said, well, a pest, you may have different populations. So depending if you do it at your national level where you know your population, but if you do it at regional level, you may have to involve more population, more differences. Also, when you do a TPS, you also have to look at, will the reagent be available to everybody? And I think we'll have some we have some contribution from TPS on that. The time constraints, maybe your NPPO just told you, well, next year in the survey, I want to plan a survey on this pest. And if you could give me some advice about the tests that could be used, then you may have to organize a test performance study because you want to have information, but then you have a deadline. You know that everything has to be finished before the survey campaign starts. And you also have constraints. For example, if you have recurring interception at the border and you're really questioned and you want to see how the test is done in different places and how the test performs with involving more labs, then you also have you may have a big time constraint there. So these are just examples, and I'm sure that those who organize TPS may have further reason to contribute there. So then we come to the next step of the process, which is criteria and selection of the participants. So what is important is that you should have acceptance criteria for your participants. We will not insist enough on the fact that one very important criteria to participate in a TPS is that you should be proficient with the test. So that's really important. It's not like a proficiency test. That's the difference. You're evaluating the test. So you need to have labs which are proficient. First, also important element, do you have the equipment available to run the test? That will be part in the TPS. So as the organizer, you need to check that the participant has the equipment, is proficient, will commit to perform the TPS. That's for the participant. And will you need to limit the number of participants? So is there potentially a problem of availability with material that you have to send around? So in this case, you may need to limit the number of participants. How broad do you want your representation of countries to be in your TPS? So do you want to give priority to laboratories coming from different countries? You also think about all the documentation you will need to prepare. So you need to provide to the participant very clear information and instruction, clear information on the timeline, clear information on how to handle the samples, how to process them, etc. And you also need to think about how will I put codes to the samples. So you need to prepare all of that. And really you need to consider all the constraints that will apply to you when you are looking at who can I select for participation to my test performance study. And so now, Charlotte, over to you.
Yes, so now we are going to the material, and so you will have to define clearly what you need as material and as reference material. So you will have to describe the requirements. So for the material, how you will produce it, how you will check its quality, and how you will store it. And so of course you have to think about the type of material you want to use. So is it going to be larvae, adult spore, plant extract, and so on. What kind of diversity of material you will need to provide to the participants, and how much you will need. Of course, how much also depends on the number of participants that you will have in the TPS. So the different steps that we are describing, they are not independent. You have to think about them globally. You have to think about how you will get the material and how you will store it. And you have to provide description of the target or of the characteristic of interest, which also include information on what the participants are going to have to identify or measures or test in the interlaboratory comparisons. So for example, will they have to look at specific morphological feature of the pest? Will they have to count bacteria or assess the severity of some symptoms? Will they have to look for the presence or absence of DNA? So this kind of information. And finally, you also have to describe the values or the characteristics that, or both, that you expect from the participant for the test samples. So all these different elements that will be further described in 2 webinars of the series. So the 4th webinar, which will be specifically about the preparation and dispatch of the samples, and also the 5th webinar, which will be specific to the production of reference material. So then you will have to think about how you will prepare and dispatch the samples. And you will have to think about the risks that preparation and dispatch can have, such as the risk of delays and the loss of samples. So when you prepare the sample, you first have to think about the number of samples that you will have to prepare. And of course, this depends on the objective of TPS and also on the performance criteria that you will want to analyze. So for example, if you want to analyze analytical sensitivity, you will have to include in your panel of samples different dilutions of your pest. So clearly, the samples that you will include in your panel are linked to the objectives of the TPS, and you have to think about it in advance. The number of samples that you will have to prepare also depends on the number of participants. So here as well, another link to a previous step. You will have to think about regulatory requirements. So what are the requirements for the participants to perform the TPS, if there are some? So for example, maybe they have to work, if you work with quarantine pathogens, then it means that the participants have to be able to handle such material and work with such material. And this is something you have to plan in advance. You also have to think about regulation for transport. So if you work with quarantine organism and you have to send it to another laboratories, laboratories will need a letter of authorization, and this might take time. So if you don't want to have delays, you have to think about this in advance and plan accordingly. Maybe also some samples, if you are working with some countries that have a specific regulation, maybe you won't be able, for example, to ship your samples on dry ice. So for example, for some TPS, they wanted to ship sample in Australia, if I remember well, but they could not ship them on dry ice. So they had to think about alternatives and how they would do it to ship the material, the samples there. Of course, then you will have to describe how you will test or measures homogeneity and stability of the samples, which is very important characteristics of the samples that you will have to prepare, and when you will have to make those measurements. So of course, this depends on also on when you will send the samples to the participants and when they will receive it and when they will run the analysis. And finally, you have to think about how you will code and label your samples. So you will have to describe the procedure to be used for the coding and labeling of the samples and to randomly assign a number of samples to the laboratories. So this will be further discussed in the webinar on the preparation and dispatch of samples. Next slide, please. So yes, and so just to mention that on the first steps of the TPS plan, so which regard the organization of test performance studies, there will be a practical training session mid-March. And just to let you know that the deadline to register to these activities is next Monday. So if you are interested, please do register today or by Monday. Next slide. Yes. So then the participants, they will run the analysis, they will run the evaluation, and then they will send you back the results. So regarding these steps, you really have to think about the methodology that you want to use to analyze the results of the TPS. So it includes, of course, for example, how will you, from the result, assess the proficiency of the participants? And actually, this is related to data exclusion. So all the data that you received, do you want to take all of them into account or not? And how will you do this? So what will be your criteria for excluding some of the data sets? How will you do to check that the controls are valid? And so that you can think that the results that you got are also valid. So yes, so you will need also to describe the statistical analysis that you will use, and you also will have to describe the information that you want the participants to report to you about how they perform the tests. So for example, is it really important for you, test performance studies, that you know the type of equipment that you use, the deviation that they have made compared to the instruction that you send to them, if they have any problem during execution, and this kind of information. And finally, yeah, sorry. Yeah, so a description of the procedure to be followed if the same sample is consistently not giving the expected results during the interlaboratory comparison. So we come back to a little bit the analysis of the results and how you will tackle the problem, how you will tackle it if you face a problem during a risk analysis. And I think we have very nice examples of problems that appear during risk analysis and that TPS organizer have to solve. So maybe Natasha will be able to tell us a bit more about this later. So for the result analysis, so there are different activities that are planned within webinar series. So the 6th webinar will be on how to tackle the analysis of TPS results. And we will discuss a lot about how to exclude the data sets, how to identify outliers. Then there will be videos as well on how to calculate performance characteristics of a test and get useful information from your validation data by the statistical analysis. These videos will be released on the weekend of the 22nd. And after we release this video, we will have a webinar which will be a Q&A session on the statistical analysis of the TPS plan. So if you have questions about the small videos that have been released, you can connect, and then you will have the opportunity to discuss with the people from Work Package 2, which worked a lot on the analysis and the statistical analysis of the TPS results. And finally, there will also be a practical training session on how to analyze the results. And here the deadline for registration is the 1st of March, so you have a bit longer to register, but if you are interested, please do so. And the last webinar of the series will be an example from the organization of the TPS to the analysis of the results. So the example will be the one of the tomato brown rugose fruit virus. So if you want to have an overview of the whole process with an example, you can watch this webinar. Finally, in the planning phase, you will have to think about how you will want to disseminate and communicate about the results of your TPS. So of course, you will have to do a TPS report, and you will have to think about how this report will look like and how you will send it to the participants. You can also disseminate your results through presentation and by sharing the data on database. So for example, the EPO database on diagnostic expertise. And I guess that here, the main decision that you will have to think about is whether you want to give full access to all of the information of the TPS, or if you want to decide on a restricted access to the data. So with this, I think we went to all the different steps of the TPS plan. So we would like, before we go to the videos, interviews of TPS organizer, we would like to ask you some questions. So according to you, in which step of the process did TPS organizer encounter major difficulty while preparing the TPS plan? So I will launch the poll. So was it more the capability of the organizer, the scope of the TPS or the selection of the tests, how to get the material, how to prepare and dispatch samples, or was it more in the analysis of the TPS results? So 60% of the people have voted. I will wait a bit longer.
I think even if you've not organized a TPS yourself, from the presentation we made, what do you think is also the difficulty? So listening, what would you figure out as difficulties?
So we have 75 of the people that have answered, and it's not changing. So I will close the poll and share the results. So 9% said the capability of the organizer, 13% the scope of the TPS or selection of tests, 34% how to get the material, 21% how to prepare and dispatch samples, and 23% regarding the analysis of TPS results. So at the end of the interviews, we will make a quick summary of what TPS organizers of VALITEST had to face as difficulty, and we will compare to these results. And we have another poll that we would like you to answer is what is the first document that you would use if you had to plan a TPS? So you have different options, the EPO standard PM7122, checklist developed internally for proficiency tests, a checklist developed internally for test performance studies, or a checklist developed internally for validation studies that you may carry out in your institutes or any other. So now again, about 75% of the people have answered, and it's not changing so much. So I will close the poll and share the results. So 65% of the people said they would use first the EPO standard PM7122. So I guess, Françoise, we are very happy to see this. And then 6% said they would use a checklist developed internally for proficiency tests or developed internally for validation studies. And 18% said that they would use a checklist developed internally for TPS. And 4% said other document. So I will hide this. And now, yeah, I give you the floor, maybe Françoise.
So yeah, so we are now, while Charlotte is launching the video, so you will see by order of appearance. So Natasha Mekle from NIP, Tania Dreo, Jean-Philippe Renvoisé from ANSES France, Tom Rijmakers from the Netherlands, NVWA, Monica, who is with us today, like Natasha, so from Unito, Italy, Marta Luigi from CREA, Anne-Marie Schaap from ANSES, and Catherine Harrison, who is also with us today from FERA. So now, Charlotte, you can launch so should I stop the presentation? Or you can take over?
So the video will start in a moment. What was the major difficulty you encountered when planning the TPS, and how did you circumvent this difficulty?
So in my case, this was how to find person that can help me with that since for such kind of work a certain expertise is needed. So personnel in our lab with that required expertise is limited and occupied with several activities. We were lucky to find one who learned quickly, but anyway, teaching someone takes enormous time.
I would say that the biggest difficulty were time constraints at the beginning of the project. We organized 2 TPS studies at the very beginning, having to send material out in March. And at the beginning, there was considerable overlap of our preparation process with the harmonization among the different TPS organizers. It was very useful. It improved our work a lot. But I would say that it's not for the faint-hearted. It resulted in good quality and useful results, but it can be a bit stressful.
As we started the VALITEST project in June, the main difficulty was to estimate the needs, to collect and prepare the plant material in the best conditions, and to organize the storage for later use in autumn and winter.
So the major difficulty we had within our organization was that we had some personnel changes during the organization of the TPS. And the problem is we didn't really have any redundancy set up. So we just had some people leading the project and then going away.
Just I am pausing the video because several people say that they cannot hear. So can you let us know if you cannot hear, if you have some problems to hear? Maybe you can try to relaunch. I don't know. Now it's only 3 people that said that they cannot hear. Is there more people that cannot hear? Can you please write in the chat? Yes, the video has stopped. It's intentional. I stopped it. So it seems that it's fine for some people. I think if you have a problem, you can stop. You can quit the webinar and then come back.
So it was quite difficult to get into the project because we didn't have one person leading it.
Sometimes it was not so easy to retrieve from the companies, commercial companies, the same material or the same buffer. But at the end, we managed it. At the end, we managed. And the reason is not because they were not available, but because working at the university, we have some special procedures for purchasing. So we had to, to work it around and prepare documents to show that we needed certain reagents that were necessary for a particular experiment, and to justify our request to be able to purchase from the companies that had what we needed. So that was, I would say, the main issue.
Tomato brown rugose fruit virus is an emerging virus. We were aware of specific biological features of the virus regarding the transmission by contact, the high longevity, and the high concentration of brown rugose in leaves and fruit. All this imposed procedures of high attention in handling the materials due to the high risk of contamination during virus multiplication, of course performed in quarantine conditions, and manipulation for sample panel arrangement. At the same time, few information was available. The main difficulty in organizing the TPS for brown rugose was in preliminary activities regarding the availability of target and not target isolates, the gathering information and validation data from the main database in order to identify the most suitable test that could be evaluated.
The biggest difficulty was to participate in the construction of the documentation to harmonize the different TPS and at the same time to carry out the technical part, selecting the test, carrying the preliminary test, building the TPS, writing its documentation before preparing and validating the samples.
I think the biggest difficulty we had for the Fusarium circinatum TPS was that we struggled to get cultures that were verified and also could be distributed in a TPS. So some culture collections wouldn't let you distribute cultures in a test performance study. So that was our first challenge.
What document did you use to cover all aspects of a TPS plan?
My main checklist, if I may say so, were two EPO standards. These were PM 7122, and so this is about the guidelines for the organization of interlaboratory comparisons. And also I use PM 798. This is the guidelines about the specific requirements. And of course, I used also the knowledge from the first round of TPS done in the frame of VALITEST.
In fact, in ANSES laboratories, we are used to organize interlaboratory proficiency tests, some of them under the cover of our accreditation. And even if there are important differences in terms of organization between a proficiency test and a TPS, the fact that we are used to organize a proficiency test was very helpful, and we only have to adapt our documentation to the specific case of TPS, but we already had a solid basis.
The checklist is essential. We have also made a retro planning from the planned sending date of panel in order to fix the schedule of action to be carried out. And we often made a point to check the progress and to make sure we hadn't forgotten anything.
Yes, we had a Gantt chart and we had several checklists for different aspects of the planning. So we had checklists for progressing methods through the laboratory, as well as checklists when we were coming to dispatching samples. That was the most complex part, I think, for requiring additional checking. We had some participants didn't do all of the tests, and there were different types of samples for different methods. So we had to keep track of exactly which participant needed which types of samples, and also ensure that they got the correct set of paperwork. Everything was checked by at least 2 people as well.
According to your experience, did you identify specific items that should be included in the preparation of the TPS plan and that were lacking or could have been improved?
We have had a very detailed plan. What we had to do is, of course, collect plant material well in advance because of the seasonality of our samples. So although we were sending samples in March, we had to, of course, collect the material the previous season. Of course, this is something that you cannot really, you don't have much maneuvering space within a project that is very tightly packed. But if we had to do it again, we would probably extend the preparation of the TPS across a longer period for this reason.
I would advise to at least, for example, if you are organizing a TPS, to have at least 2 people working on it and being aware of what is going on. So build in some redundancy. So when someone leaves for whatever reason, you can continue without any difficulty.
I would say that each laboratory really has to prepare its own checklist because each situation may have different conditions that need attention.
The detail on the composition of sample panel should be improved in the way that inclusion or exclusion of a specific sample can affect the evaluation of performance. So every sample that must be included should be carefully think before preparing the TPS.
As already said, the most important thing for me is to put in relation objectives of the TPS, appropriate choice of the samples and data analysis. All this before the beginning of the TPS.
So this is the end of the video. So Françoise, if you can share
Share my
again
screen
your
anymore.
presentation.
Okay, yeah. Show my screen. So which one?
Yeah, it's okay.
It's okay. So, yeah, I had forgotten Aude from who we last saw in the video. So as you have seen with the experience reported by the TPS organizer, so the capacity, so they mentioned the expertise in a TPS plan, the fact that you may have personal turnover and that you need to ensure this issue of personnel can be important. The internal procurement procedure, which in one case were overcome, but created some delays. When you select the best, yes, the lack of information and validation data. The needs regarding the material, there was not much reported regarding the participants, but regarding the material, clearly obtain the material, the region, being able to use the purchased material in collection in a TPS was an issue. So if we come back to your answers, we can see that we can find some of the difficulties that you have highlighted clearly there. The handling procedure. And regarding the issue of the time constraints, so the period on when you need to get your samples compared to when you need to do your TPS. And in the case of the VALITEST, there was an overlap between the preparation of the TPS and the harmonization process in the documentation that was done in the framework of VALITEST. So so now we would like to launch another poll. So I think you will have to stay with us for a few moments. Charlotte, did I?
Yeah, this poll was already launched. It was just to show that there was no really wrong answers. I mean, all answers were correct, but and some TPS organizers used, of course, PM7122, but we can see that Also, organizer use checklists that they've developed internally for PT or TPS or validation studies and other documents. So, but what we can say, and maybe I will get you the floor, is that specific documents were developed in the framework of VALITEST.
Yeah, so this is an illustration on how there was a time frame in VALITEST, and they used a lot the Gantt chart to mark the different steps, like listing the pathogen, which was already done in round 1, because when the proposal was submitted, the list of pests was already selected. But then we had a second round regarding for the second round there was a need to select the pathogens. Then you have time for the literature search, collecting your plant material and your target organism, the criteria, the selection of tests for validation, to get the chemicals, to get the validation of tests. So all these steps are described. I think because time is running, I'm not going to go into the detail, but for each of the steps, you can see that there was a calendar, and I think it's Catherine who mentioned, or Anne-Marie who mentioned the counting back. So this is really important, and you really need to have this planning in place for your TPS. So a real poll this time, not just a reminder of another poll, sorry. So would you be interested in having access to the documents developed in the framework of VALITEST? So that's a quick question. So
yes, as expected, we have a majority of people that are saying yes.
So there will be different ways for sharing, and of course we will keep people informed about this. And for example, there is a book which is planned to describe the TPS based also on examples. So the advice from the TPS organizer: so preparation and planning takes time. You need to have several people involved. You need to have your checklists, and you really need to keep in mind the objective of your TPS when you plan the choice of the samples and the data analysis. So now I'm sorry, but I think it's important to still have some time for questions, so we can go a little bit over the hour if you have questions. So I would like the TPS organizers to join us and switch on their cameras. And please ask questions. If you want to take the floor, please just let us know, because these people are the experts. We've just been the replacement for the presentation, but they are the one who have the expertise.
So you can ask your question in the chat, but you can also raise your hands and we will give you the floor if you want to ask the question. So yes, so we have a hand raised. So Amandine Levent, so go ahead. You have to unmute yourself. Amandine, you're self-muted, but you should be able to unmute yourself by clicking on the mic, who should turn green.
The hand was raised 6 minutes ago, so.
Oh yeah, okay.
So
it's too bad because we have no questions or hand raised. So do not hesitate to take
Charlotte,
the floor.
it seems that there is somebody who tries to say something. I heard the voice kind of far, but.
So Amandine is saying that she has a problem with her microphone. So Amandine, maybe you can ask your question in the chat. So it was a question regarding homogeneity and stability, but I don't have the question. So maybe Natasha, Monica, and Catherine, you would like to say something? Oh, we have a question. So the question, is there a direct platform? Is there a direct platform for finding participants? Is it an open call through VALITEST, or the organizers contact potential participants directly? So I will put the question in the chat. So I guess that Natasha, Monica, and Catherine, you can answer for VALITEST how it was done.
So maybe I can answer very quickly that actually there is no platform how to find the participants. What we use is that we send the message to people that we know from EPO panels, from our previous organization of proficiency tests and other contacts. So also with the help of EPO, we actually distributed this to the many potential participants.
Same was for us. We chose the participant, of course, among the VALITEST project partners, and we added other people that we knew or that we retrieved through other sources like Natasha did. But there is no platform.
So now we have the question from Amandine. So if you want to validate a new test compared to an alternative one, do you have to do the homogeneity and stability analysis using the two tests, or can this be done only with the reference test? I'm sorry, Amandine, I put it in EPO wording.
I didn't find myself in this situation because we had no reference test for the pathogen that we have organized. If I had, I would do both for homogeneity and stability.
I must say that we did homogeneity and stability tests with all tests included in TPS.
Yes, the same for us.
The matrix were different, prepared and so on, that's why.
Yes, we did homogeneity testing and stability testing across all tests so that you had exactly the same material, so you could compare your tests against the set criteria so that there was no difference between what you're using on every test. It has to be the same across every test.
And Amandine, if you want to know more on this, I guess you can join the webinar that is specific to the preparation of samples and dispatch of samples. I guess this will also be discussed there. So I don't see any other question. Yeah, no, so I would like to thank Françoise and of course Natasha, Monica and Catherine for joining us today. And I would like, of course, to thank you all as well for your participation. There will be a satisfactory survey at the end of the webinar and or in the follow-up email. So it is really short, so do not hesitate to provide your feedback to help us improve the webinar experience. I am really sorry that some of you got some troubles with the video interviews, but the video will be available on our website as well, so you can watch the interviews again, and we will also put the presentation and a link to the recording of the webinar. So the next webinar will be next Wednesday, and it will be about the selection of the test and the associated documents. So if you're interested to know more about how you can select the test for TPS you want to organize, then you can join this webinar. And do not forget to also register to the practical training activities. So the one on how to organize test performance studies, the deadline is next Monday. So please, once this webinar ends, go and register. Thank you all, and see you another time.